Hi everyone, welcome back to my single-cell scATAC-seq video tutorials. Today, I'm going to show you how to integrate scATAC-seq data with analyzed RNA-seq data. Integrating with RNA-seq data is the best way to name all the cell types and cell subclusters for your scATAC-seq data. So in my previous video tutorials, we analyzed the PBMC dataset, and also we predicted the gene activities. If we have a look at the PBMC dataset, you can see we have two assays, the peaks and the RNA. So the predicted RNA expression is important to integrate with single-cell RNA sequencing dataset. So now we can read the RNA-seq data in. You can see we read it as PBMC RNA. So let's have a look at the cell clusters in the scATAC-seq data and also cell clusters in the RNA-seq dataset. So we can look at the cell clusters together. Let's run. We can zoom in. You can see on the left-hand side are the cell clusters from the scATAC-seq data. We have 15 cell clusters, and on the right-hand side are the cell clusters from the RNA-seq data. You can see in the RNA-seq data, all the cell clusters are named from previous analysis. If you want to perform the same analysis, you can download the RNA dataset from the online tutorial from here. So now we have both datasets. We can perform integration, use the cell clusters from RNA dataset to enable cell clusters in scATAC dataset. The first thing we need to do is to find the transfer anchors. Use the RNA dataset as a reference dataset to enable the scATAC-seq dataset. We know when they did the analysis for the RNA dataset, the reduction method is CCA. So we can run find the transfer anchors to identify anchors between the scATAC-seq data and the RNA-seq dataset. Let's run. Okay, so we have the transfer anchors now. Then we can use the transfer anchors to transfer the cell labeling from the RNA-seq data to scATAC-seq data. So we just need to perform the transfer data. Use the transfer anchors. So we named it as predict labels. If we have a look at the predict labels, you can see now each cell has a predicted name. So let's add the predict labels as metadata to the PBMC. Before we do that, we can have a look at the metadata. You can see now we don't have the cell type names for each cell. If we add the predict labels as metadata for the PBMC object, then we can have a look at the metadata again. Now you can see the cluster name has been given to each cell. in the column of predicted ID. So all the cell canisters in the scATAC-seq data have been labeled. So now we can go back to visualize the cell canisters. So we can use the dim plot function to plot the RNA data set as plot 1, scATAC-seq data as plot 2. Now we can visualize them together. Let's zoom in. You can see now for the scATAC-seq dataset, all the cell canisters are labeled. Well, no, the pattern of cell canistering is different from the RNA-seq dataset, but you can find the same cell canisters from the scATAC-seq dataset as the RNA-seq dataset. You can see we have the B cell canisters here in the RNA-seq dataset. B cell canisters are here in the scATAC-seq data. Monocyte and the dendritic cells here in the scATAC-seq data are monocyte and dendritic cells. You can see the cell canisters in the RNA-seq dataset for the T cells. Here are all the cell canisters for the scATAC-seq data. So by integrating scATAC-seq data with analyzed RNA-seq dataset, we can label all the cell canisters. You can see at the moment we can find the same cell canisters between RNA-seq and scATAC-seq, but the pattern of all the cell canisters are different. So we can perform co-embedding for the RNA-seq data with scATAC-seq data to make the cell canisters have the same pattern. So let's perform the co-embedding analysis. So we are going to merge the scATAC dataset with the RNA dataset. So before we do that, we want to label all the cells in the scATAC dataset as scATAC and all the cells in the RNA dataset as RNA. So we can create a column as sequencing technology for scATAC dataset, and we name it as scATAC. So if we have looked at PBMC metadata, you can see we have a column to label all the cells as scATAC. We can do the same for the RNA dataset. Let's have a look at the metadata for the RNA dataset. You can see we have a tag column. All the cells are labeled as RNA. Now we can perform co-embedding. Once again, we are going to use the names for the cell canisters in the RNA-seq dataset. We know when we perform the single-cell RNA sequencing dataset, we use the variable features to canist the cells. So we can just use the variable features as the reference data from the RNA assay. Then we can get the assay data for the variable features from the RNA dataset. So we can transfer the data from the RNA to the scATAC-seq data again, and add the RNA data matrix into scATAC-seq data. Now we can merge both datasets and we name it as co-embed. So now we only have one object. We can have a look at the object. You can see it contains both RNA and scATAC assay data. So now we can run standard analysis to visualize the cell clusters. First we run data scaling, then run PCA, and finally run UMAP. Now we can visualize the cell types. We can group the cells by ATAC and RNA, and also we can group the cells by cell types. So let's run. So now we are ready to look the cell clusters. Okay, so my plot windows is too small. Let's run it again. So we can zoom in. You can see now the cell clusters from scATAC-seq and RNA-seq have the same projecting pattern, and only these cell clusters from the scATAC-seq data is not presented in the RNA-seq dataset. So on the right hand side, we can see the name for all the cell clusters. So I finished today's demonstration. We performed the ATAC dataset and the RNA-seq dataset integration, and also we performed the dataset co-embedding. If you haven't subscribed my channel, please click the subscribe button after you watched my video. Thank you. See you next time.